Hello. In this lecture, we're going to speak about brain circuits, how they develop, and how they connect before birth and after birth. So first, we need to keep in mind that in order for you to build brain circuits, you have to start with having the units, the building blocks already. And the building blocks to build brain circuits are the neurons, and these neurons have to be able to transmit signals to other neurons. So in order to do so, they have to form synapses. And once you have synapses, then you have the elemental or the elementary foundation of a network. So we're going to speak a little bit about synaptic formation and how synapses are eliminated. And we're going to speak after that about the formation of circuits and brain networks. So the synapse is the basic processing unit of the information, where in order for you to transmit a signal from one location to another, you have to establish a synaptic connection where one neuron transmits the signal to the next one. And this process begins in early fetal development, and it is guided by three processes, how to form synapses. So here you can see one neuron and other neurons that synapse with it so that they can communicate. First process in synaptic formation is axonal path finding. So first, the axons of neurons, these endings, if you look at, here's the body, here is the axon, and the axon terminals. These transmit signals to other neurons. So the axon terminals have to extend and to look and search for targets. And in that process, they navigate because the brain has billions and billions of neurons and neuroglia and all kinds of structures, and each little axon has to navigate its way and to grow in a way as to bring it closer to its target, and then to develop and extend these synaptic terminals so it can connect on different targets. And these targets could be dendrites of other cells, cell bodies of other cells, or could be axons of other neurons, and so on. There is a lot of competition. Just think about like sperm, as millions and millions of sperm race toward that egg to fertilize it. Generally, one is gonna make it. And that one, as soon as it touches, everything now changes. For axons, the same way. They all compete for targets so that they can converge on and connect with. And there are molecular markers that molecules have that guide them to their targets, that guide them to neurons that they know that they can communicate with, whether to transmit signals with or to mostly that is what determines what comes after actually the axon itself, as it searches, and then it determines the next cells that it will connect with. So by doing so, now neurons also realize or recognize each other by activity. So when there's something that happens, an activity or some kind of, let's say you hear lightning, and that startles you. The process obviously occurs because of neural responses in your nervous system. So at that moment where you hear the auditory stimulus, all of these neurons spontaneously begin to fire in response to that. However, only neurons who are specialized in responding to this, that stimulus are going to just, to fire. And as they do so, then they recognize each other and they release signals and they kind of reach toward each other and they connect. And this is where the saying goes, neurons that fire together wire together. So axonal path finding, and this is when axons determine, identify, and locate their targets on other cells, and they connect with them. And once these axons are formed, then the nervous system then begins to to kind of receive and send signals, and that's a major part of fetal development. Synaptic differentiation is the second factor, the second determiner of how synapses form. So once the axons have synapsed with their targets, then the presynaptic terminals, the neuron that is sending, and it has an axon, it has these synaptic terminals. These components that are called the presynaptic terminals and the postsynaptic dendrites, now they become kind of differentiated, they are specialized, and they kind of together they start to receive. I am the sender, I am the receiver, and they continue to work this way together. So synaptic maturation occurs immediately after the synapses are formed. Once a synaptic connection is formed, then it starts to mature with stimulation and continuous firing. Then we have the process of synaptic pruning, and we'll speak about it in a few minutes more. But generally, it is a process that goes along with synaptic formation. So I'll explain it again to you in a few minutes. We'll come back to these images. But in any case, experience, which means activity, which means stimulation through sensory modalities, that's the only way through which we learn. So the stimulation is going to cause two types of alterations in brain structure and the nervous system in general. Number one, short-term physiological alterations on the synaptic level. Physiological means functional, like the functions of these neurons, how they behave. These functions are going to be modified on the spot, and that is short-term. Now, these short-term accumulation of these short-term alterations in physiology, how you use the network or how you use the synapse, that leads eventually to long-term alterations on the anatomic level. Like major areas then will become wired in a certain way, or will become connected in a certain way. So short-term functional alterations lead to long-term alterations on the anatomic level. And you have millions and millions of neurons. Once they change behavior, then you are going to start to have large areas changing connectivity and changing their anatomy. So for these changes to occur, you have to have repeated stimulation. You have to kind of keep using your sensory channels. So the short-term physiological modifications include, number one, strengthening synapses or weakening synapses. If a synapse is not being used or is not being used well or is not connected correctly, then the nervous system is going to dismantle it. It will basically withdraw the synaptic terminals, and once you withdraw, the receiving cell is going to kind of wait and wait and wait. There's nothing that's happening, and then it's going to also withdraw, and it will dismantle its part that was making the connections, which is synaptic. Most of the time it is dendritic spines. So the other way to for example, you either strengthen or you weaken the synaptic connectivity in the short term, and also elevating or decreasing the effectiveness of synaptic transmission. When there is, let's say, you meet someone at a party, you hear their name for the first time. Now, that is kind of, you heard the name once and it is labile now. It might be forgotten, but if a few minutes later you speak and you hear their name again, and 5 minutes later you keep hearing their name, the nervous system determines if you are hearing it so frequently, it is important, and it's going to build a network so that it can hold on to that name. And as long as you keep being exposed to it by hearing it, by seeing it written and so on, or seeing the person, that synaptic connection is going to be enhanced. It will be, the function of it is going to be refined so that you will use it for the long term. And the opposite is true. If you only meet the person once, they say their name, and then 6 months or a year go by, and you just look at, you do not remember the name, given that, say, it's a new name you had never heard before or associated with someone else, that connection that had that name initially is not going to be used again, and it will be just dismantled. So these short-term physiological modifications, whether by eliminating or strengthening synapses, or by increasing their performance or decreasing their performance, that when you have billions and billions of neurons doing that, then you are going to have large-scale alterations on the anatomic level beyond the cellular level. So again, cellular changes drive larger structural changes. But what I want to emphasize here is that these things are real, that what you learn, what you get through your environment actually changes and shapes your brain. These are not metaphorical things. These are real changes. So here you can see the dendrites, how they are spreading like the branches of a tree, and all these pathways and how they connect. So the connectivity, again, is always, not always, I'll tell you about the situation later, but the main trigger for strengthening and influencing the connectivity is experience from the environment. The long-term modifications that result from these short-term changes include anatomic modification to the circuits that are involved, that include these little synaptic connections, because a circuit is a huge area. It's pathways and that connect many areas together. So these modifications that involve the circuit include, number one, pruning of pre-existing synapses, and also sometimes growing new synapses. And so you have dendrites. You can grow a dendrite, or you grow synaptic spines on dendrites, so you can make more connections, like these, for example. And the dendritic spines are the sites for synaptic connections. So again, you may dismantle whole synapses and develop new ones. Also, another way for these modifications to occur on a larger level is the development or the elimination of dendritic spines themselves, as you could see here. So if you have more, it means you can connect with more neurons that will send their axon terminals to make connections. So these changes, I mean, these dendritic spines are in the billions, and when you expand them, of course, they'll add to an increase in brain volume. How the synaptic connections mature? Initially, when the synapse, is being formed, initially, you want the synapse, you want it, in order to activate it and to start to build pathways, you have to make it fire, like priming it. It's like a pump. You start the pump once, twice, there's nothing, but you have to keep it try, and then eventually it starts to pump. So first, when, for example, we have over here the cortex. That's a very thin layer, the very top layer of that surrounds the brain. And then you have lower structures like brain stem areas and lower, structures here. And then these structures are connected with your peripheral system, like with your brains, with your spinal cords, with your arms, with the lower parts of the body. And these enable you to feel sensation. But now we're speaking about a situation where these lower brain structures haven't yet connected with the cortical targets that actually enable movement and kind of process information. So how do you do that if you don't have environmental stimulation yet? Because, for example, the auditory system is not developed, or the visual system, and so on. How do you begin to prime and to get the circuits, the synapses and the circuits to actually fire initially? You can think about it this way. It's almost like when you go and buy a lawnmower. You might not have, but so sometimes they give you a little bit of oil and maybe even a little bit of gas, to if it's not electric, and just to keep it going for the first time, and then you take care of it. So in that case, before sensory channels and mechanisms are connected with the cortex, and the cortical areas that send and receive signals from them, you have in the cortex and below the cortex. The cortex itself is called, in that after birth, in that part, it's called the cortical plate, that thickness of it. Beneath it, there's another layer that is basically the white matter. It is narrow a little bit at the beginning, and we call it the subcortical plate. So there are autonomic nervous system functions, spontaneous things that are genetically determined. For example, like how the heart beats. That is, a system that is run by itself, autonomic. Your stomach digestion, you have no control over it. So in the cerebral cortex and the subcortical plate, there are autonomic nervous system mechanisms that actually fire spontaneously on their own initially. And the purpose of that is to stimulate these networks so that they can fire, and then as they fire, they continue to, carve their path and to go and find targets and connect with these targets. Just like a river forming its path. So these autonomic firings continue so that they can drive the connectivity of a network. And after that, once now you have connected the network, you are able to receive signals and to send signals between the cortex and lower structures of the brain and the nervous system, and to receive sensory signals also. Sensory signals develop first. So you get sensation from the body, the skin and the inside of the body and outside and so on, so that then they are the first to connect onto the cortex. And as a result, the baby, again, as we discussed in a previous class, around 14 weeks or so, the baby had the entire body is covered with sensory endings so that the baby can feel everything on the body and probably within the body as well. So once these synapses are formed, the receptor channels are redistributed and are concentrated at the site of the synapse now, so that you send firing, and then it just induces changes in the membrane of the cell that is receiving the signals, and as a result, it's going to, based on the number of action potentials it's getting, it is going to, and the neurotransmitter and so on, it's going to put the adequate number of channels that will open and close to take in ions that transmit signals. So, and that's the normal process. The neurons increase and decrease the number of these channels as you are performing. So these synapses mature with continuous stimulation. Hearing that name over and over and over and over is going to make it now well established. You do not have to wait. You are going to just retrieve it immediately, like you retrieve anything very fast. It becomes automatic and spontaneous. Are also, if there is something that blocks the sensory stimulation from reaching the cortex, that is going to lead to weakening of these synapses, and if that continues for a long time, it is going to produce abnormality in the connectivity of the network or the circuit. And as we discussed before, if these blockages occur or interruptions occur during the critical periods, the resulting problem might not be reversed. So we wonder, how do these synapses, the synapses themselves, the neuron that is sending a signal and the one that is receiving a signal, these are not exactly touching. There's a very small gap in between them, of course, so that the neurotransmitters and the chemicals can flow, as you could see in the image here. But what holds them together? There's a whole bunch of molecules called adhesion molecules to make the synapse, two parts of the synapse remain together. As you can see, some here are in the picture. There are also there's a number of molecules called synapsin 1, synapsin 2, and so on, that are also, they contribute to making that, holding the two parts of the synapse together. And with more experience, you have stronger connectivity that will make the memory more lasting on that synapse. And also you need glial cells so that they are necessary for forming the synapses as well. This is kind of a representation of synapsin one. It is called synapsin because of the synapse, and you can see how it is structured, so it contributes to the connectivity to hold synapses together. So now that we touched upon the development, the cellular components that enable us to have the building blocks to form networks and circuits. Now we'll speak about how these circuits and how these networks actually develop. Typically, a circuit tends to have less locations, like a smaller than a network. However, in many cases, we tend to say network and circuit kind of together. One can function instead of the other. This is a study, and it is a review based on many studies, or basically a lot of research that focuses on connectivity and pathway formation. And the study, I will make it available for you online. And also, I would like to remind you to kind of listen to this lecture for about a half an hour and put it off. Pause it, do something else, and then come back to it, because it could be quite tedious. I try to put it in one video so that it will be more convenient instead of having more than one video. But you can do it this way. You can just pause it and listen or watch it for a half an hour and then come back to it. Brain architecture at birth. The basic layout of the brain is formed by the end of pregnancy. So full-term pregnancy is about 40 to 42 weeks. So by the end of the third trimester, the basic brain architecture is there. So for example, you have a right hemisphere, you have a left hemisphere, you have a temporal cortex, and you have all these basic structures, but when they are going to expand and they're going to become more sophisticated as they develop. You could see in the image here, 27 weeks, the brain basically the surface is just about smooth. And then by the 30th week, you are going to have distinct gyri and sulci, but they are not deep enough, the fissures or sulci, and the gyri aren't elevated enough. That plays a major role in function, because you need to have well-developed gyri, like little mountains and valleys, so that you can function well. And some studies with autism, they show certain areas are not as deep in terms of sulci, and some of the gyri are not as prominent, are not as high, which means that there are areas of the cortex that are thinner than normal. So as you can see, gyrification, the process of forming gyri, increases with development as you go, and it continues after birth. So the large-scale connections are in place at birth. So once the baby is born, now, given that everything is normal, the entire layout is formed, and the basic large-scale connections of the circuits are there. But we need to remember all the time that these circuits are genetically driven, and they are tentative. They are modifiable, highly plastic, and the environment is going to sculpt them, is going to shape them in different ways to make the person fit for that environment and for the body that they are living in. So again, at the beginning, brain connectivity is stimulated, is driven by genetic factors because, as I mentioned before, you need to have stimulation, you need to have these neurons fire, keep flashing and firing. As long as they are flashing and firing, they are connecting and transmitting and receiving information. But how do you do that if you don't have your senses yet connected with the cortex? Then you have the genetic, genetic factors are going to determine that, and even will give you some firing initially to prime the circuits to actually make them go from point A to B. And once you have reached, now you are on your own and you just keep going. It's almost like I was watching a penguin show, in the south, the Antarctic, and they said that the male penguin stays for about I think 40 days, or I don't remember. Don't take what I say in the duration for granted, but it's a long time staying there in the cold and suffering and all of this, and no food. But once the egg hatches, then at some point they feed that baby one bite that they had stored for all this long, so that it can get the baby kind of sustained for this period of time until the father, I think, or the mother comes back. I don't remember, but just the focus is that initially the nervous system induces its own autonomic firing to activate so that the circuits can connect. And once they connect, then they depend on on stimulation from the environment, because now the senses are connected. If there are cases when we have developmental abnormalities in the connectivity, and this would result usually from, number one, genetic causes, that the DNA is not sequenced, all right, and there are genetic problems, say for example familial or whatever, but genetically based. And they also could result from epigenetic modifications to the DNA, and they also could result from a combination of genetic and epigenetic. So, for example, an example of a combination is, for example, when you have a gene that is dormant, and in order for it to express itself, it is going to need some environmental stimulation. So the environment can positively make a gene express itself, or can negatively affect a gene and make it suppressed, so that gene cannot express itself. For example, the gene the genes that make it possible for us to produce new neurons in the hippocampus so that we can learn. When someone is under stress, extreme stress, these genes are suppressed. They cannot express themselves. They cannot translate. They cannot be copied or be translated so that they can make new neurons to replace the dying neurons in the amygdala and the hippocampus. And as a result, if that continues for a long time, that hippocampus could lose about 30% of its volume. The thing shrinks by 30%. So, but if you have a positive experience where the person is receiving like sensory stimulation, when the person has good social relationships, and they feel happy and adjusted and all of this, then you have an increase. Now you cause these you cause these genes to release, to express themselves even more robustly so that they can produce an increased production of the neurons that enable us to learn and to connect our learning to what we already know. During the second trimester, the connectivity it progresses remarkably, and we notice exuberant, especially like peaks in the progression of these three processes. And synaptogenesis, the formation of new synapses, and dendritic sprouting, the dendrites on the body of the neuron are sprouting and increasing in number, and axonal path finding. So the axon, as it keeps extending and then it reaches, say its target, then in the process it also branches and it strengthens, and the synaptic terminals become more mature. So these processes of synaptogenesis, dendritic sprouting, and axonal path finding, they begin prenatally during basically the second trimester, and they continue more robustly after birth because of the experience. And we know that the environment and the experience will shape 2/3 of the brain. So these three processes give rise to neural circuits. Now you have the building blocks, you have laid the foundation so that you can now get these elements and now make larger and long-range connections to establish networks and have whole areas now, connected so that you can do more with them. The first circuits that form are between the thalamus and the cortex. And when we say thalamocortical, it means here is the thalamus in the core of the brain here. And this is, it sits on top of the brainstem. These are the two sides of it, left and right. And it is the size of a bird's egg. And the two are connected. This structure serves as the connection between sensory mechanisms, like lower areas, like the rest of the body basically, and the cortex, and also it relays also motor functions as they come through. They go through also the thalamus. So the thalamus has compartments for basically every sense, except olfaction, except smell. And it does have smell. It does have smell. But the idea is that it is connected extensively with the left and the right hemispheres and the frontal parts of the brain, the back and lower and up. It is extensively connected. And it serves as a sensory gating mechanism that controls the force and the power of sensory stimuli as they come from below. It is going to either enhance them or kind of suppress them a little bit to kind of get them to make them in the ideal form before they go to the cortex. So you have now, when we say thalamocortical, it means these pathways send signals from the thalamus to the cortex above. And corticothalamic are networks that send information from the cortex down to the thalamus. So this is the difference. You might think, oh, they are the same. No, there are pathways that send down and ones that send up. So it's a bidirectional path, bidirectional connections between the thalamus and the cortex. It's like two-way streets. So initially, so these are the first networks that form. And these networks are tentative initially. They are in what they call it, rough draft mode. They can be modified and changed. And they form, once they form, then the thalamus and the cortex become more differentiated in function. Each one is going to now have, distinct areas that will start to show clustering of, connections, and then that will give rise to specialization of these areas in different functions. So these networks, thalamocortical and corticothalamic networks, they form between the post gestational week of weeks 24 and 32. That is after birth. So the thalamus is again the major sensory mechanism, and all the senses basically. I have to take this smell out. I was speaking about a different mechanism. The basal ganglia basically, and different mechanism. But so they pass through the thalamus, and it connects sensory, inputs that are coming from below to the cortex. So always sensory inputs come from outside or inside of the body and rise to the cerebral cortex. Motor commands come from above and they go down. So you could see some of these pathways, motor circuits coming down. These are corticothalamic and associative circuits. These are for integration of different sensory modalities together and sending that to the frontal part of the brain. And they are also we also have the limbic circuits, and these deal with emotions and motivation and so on. So it is extensive. This structure is extensive, and it makes sense that because of its importance, we have these networks. They form at the beginning. They form first. Just to highlight the importance of the thalamus, there is a whole journal called Thalamus, an entire journal where they publish information just dedicated, research dedicated to the thalamus. So 4 weeks after the formation of the thalamocortical circuits, sensory neurons now, including the auditory neurons and visual neurons, are able now to send signals to the cortex. Now they have made a connection and they can send signals to the cortex. So at the same time, the corpus callosum that connects the left hemisphere with the right hemisphere, that is like a bridge. The corpus callosum begins to extend from the front to the back. So from anterior segments to the posterior segments. As it keeps developing, you could see some here. So it keeps growing, connecting and connecting as you go back. So that this way it is connecting right hemisphere with left hemisphere. So as soon as it connects, it connects with neurons in the cortical and subcortical plates. As you could see here, the axons of neurons are here in the white matter, and the bodies of neurons are in the cerebral cortex. So again, I emphasize over and over that in order for neurons, for the brain to form circuits, so the nervous system to form circuits, there has to be some kind of firing. There has to be stimulation. So, and that stimulation again is autonomic initially. And these autonomic firings drive neural circuit formation before sensory stimuli are online, are functional. But once you have now this, it's like establishing a highway between one city and another. So how do you you find a way to keep going and building the pathway, but once you have connected now the pathway, now we can travel on our own. The sensory driven cortical activity that is continuous and it is continuously received gradually increases with the advancing age. After basically from the point of conception to about week 50 from conception. So you have about the middle of the third week. So by that time, by the middle of the third week, now we have these networks. They have been continuously stimulated, given everything is normal. And they are connected and they are able to fire on a consistent basis, on a continuous basis. So, and that again occurs around 2 months and a half after birth. So that again, it continues, but it starts before birth, but it continues through to about the the middle of the third month. That is a time where you are going to have, for example, the baby is able to laugh and to giggle, and the baby becomes like has a great personality that is noticeably clear to people. Look at the temperament and a lot of things that you can now understand about the baby at that time. But you could see how circuit connectivity is going to give rise to these behaviors that we see. But without that, then how could we get any behaviors or any emotions and so on? The myelination process is also important because it gives rise, it contributes to circuit formation, because a neuron that's not myelinated well is not going to function well. And the degree of insulation and is going to increase or is going to determine the level of neural transmission, how fast you can transmit signals and how effectively you can. So myelination is required for circuit formation, and it starts around the third during the third trimester before birth. So it begins with the maturation of these oligodendrocyte cells that we discussed before, that develop, that make the insulation for cells in the central nervous system. And these cells exist in the white matter, and they begin to form myelin. So generally, there's some kind of a basic pattern. The primary cortical centers are myelinated before association cortices. What does that mean? In the cortex, there are basic areas that function as elementary centers for particular functions. And we call these, usually have the word primary before them. For example, we have the primary motor cortex, the primary auditory cortex, the primary visual cortex, the primary sensory cortex or somatosensory cortex. So all of these areas, and the primary auditory association, so all of these areas that are called primary, they are specialized areas that serve as the nucleus of a network. So they have a whole circuit, but they are the engine in that circuit, in that loop. So the myelination process progresses in the primary cortical centers first, and then it goes to the association cortices. Association cortices are long pathways that connect multiple areas of the cortex. For example, in the front, there's an association, a frontal association area that connects with the back of the brain, like with the parietal cortex and the visual cortex and different areas. There is an association, auditory association cortex here, and there's a somatosensory association cortex here. There's a visual association cortex. So all of these association means integration of multiple modalities, multiple sensory and multiple cognitive modalities getting together, and you get the information merged so that you can, for example, see something, hear it, and while you are seeing it, you are dancing or doing, say, karate performance with the whole group, watching the group, listening and watching and moving your body in a coordinated way. All of that requires motor and visual and auditory integration. So also the myelination process progresses in the following manner. From caudal to rostral. Look at the brain now. Caudal means brainstem. Rostral is the high point, which is the frontal, the very, very top of the frontal part of the brain, this area. So myelination begins here first, and it keeps going on and on and on and on to the frontal. Rostral to, caudal to rostral. And then from central to peripheral means the brain and the spinal cord are myelinated. The brain and spinal cord are myelinated first, and then myelination goes to the cranial nerves, and myelination goes to the spinal nerves that come out of the spine so that they could innervate the entire body. Then also myelination progresses from the posterior areas here, that are visual cortex, and it moves forward to the anterior. So again, caudal, rostral. Rostral comes from the word rostrum, means beak of a bird, like the top of like the head in relationship to the body. And caudal means the tail. And then you have the posterior, anterior, and then you have we said central means brain and spinal cord begin to myelinate first before the other peripheral structures. So during the third trimester now, we're speaking about the late fetal period, there are pathways that connect right and left hemispheres. These are the interhemispheric pathways, and long association pathways that I mentioned. They now extend into the cortical plate, like they send their extensions to the cortex itself. So now they are connecting the two sides of the brain, of the cortex, and they are also the cerebrum, left and right, and they are also establishing long-range associations that connect front to back and left to right and different parts of the brain. So at the time that the axons are extending and connecting with their targets, at the very same time, as they increase these connections and the white matter is maturing and expanding in volume, and the gray matter is also maturing and developing, then that actually causes the brain to swell, to develop, to grow in size. Where do you get the space? Because the skull is limiting everything. So now to find the way, an effective way to do so is to fold and convolute the different, structures of the cortex, and so you can fit as much inside of the cortex as possible, and to expand the surface area of the cortex, because the cortex is what makes us as a as a species more advanced than any other species on Earth. It's the size of it, how extensive it is. So at the very same time that these axons are connecting and extending and enlarging their size, then you have the convolution and the gyrification, where the gyri start to be, more elevated, and the sulci become deeper. So postnatally, we look now after birth more in depth at the networks after birth. By the end of normal gestation, then, all major white matter tracks are in place. So white matter tracks, there are many pathways that, kind of as the gray matter here, where you have the cell bodies, and then they send down their axons down, so that they can connect with lower areas of the brain. So the structures of the newborn, the brain structures, are basically similar. Of course, not the function, but the basic layout of the brain is similar in terms of there's this area, there's this lobe, and so on. So these topography landmarks are there, and they are similar to that of the adult brain. Then you also have the macro scale white matter connections across the white matter, front to back and sideways, and all kind of various parts of that white matter. You have pathways and long-range pathways that are going to kind of to be formed by the time of birth, or by the end of pregnancy. And after that, after these macro scale white matter and long-range connections are made, and they connect the two hemispheres, and they make projections, then short-range cortical connections now begin to form. So they continue to form through the 4th postnatal month. That is 4 months after birth. So age 4 months. And you could say, for example, you could wonder, why do you have to have like highways before you have local, streets and so on? And it is possible that the highways are going to serve as pathways to bring in that stimulation that is going to bring in people, and to bring in people to build towns and things, and to bring supplies in and out and all of this. So it makes sense. So the characteristics of the postnatal period of circuit formation and development include increased integration and increased clustering or segregation of modular cortical areas. Integration always means that you have multiple functions, and then you blend them into one together, so that you can get greater things out of them. So like I said, you use your vision to guide your motor movements, to guide your auditory, you use your auditory signals to guide your motor and your vision. Like, so there's a noise, and then you turn your head. Where is that noise coming from? So you guide your vision and you guide your motor movements. So that is all integration. Multiple sensory channels converge together, and you get out of that the ability to do multiple things that are more complex. The clustering and segregation, it means that at that time you have these local areas that keep firing and firing, then they specialize and they make modular components. For example, this red area here is actually the green area. This is the motor cortex, for example. It is specialized in enabling all different parts of your body to move voluntarily. So, and the area behind it, the sensory area, that is the sensory cortex, primary sensory cortex, and that enables you to feel everything on your body, any sensation, pain, temperature, and so on. You feel it there. So if you look at every single area, you find these modular components that are specialized in particular functions. Like here, for example, about here, there's an area called Broca's area that is specialized in grammar, in syntax, and in motor activation for speech. Of course, it can't work on its own. It has to be part of a major network. So these areas at that time, they are clustered, they are segregated, they are specialized, and their shape becomes more distinct as they continue to become more and more connected with continuous stimulation. So the networks of primary functions, when you say primary functions, like I said, they are mediated by primary cortices, primary auditory cortex, primary visual cortex, primary somatosensory cortex, and so on. So these are localized in particular areas, and their functional connections are limited in these networks, the primary areas. Their functional networks at that time are limited to their homologous counterparts. For example, Broca's area, as I mentioned, and I point to the left hemisphere here, it is only found in this area, in the left hemisphere, in the inferior posterior frontal lobe here. And what we are saying is that at this early stage in development, this area is primarily, it is, yes, identified, you can see it, the shape and the focus of it, but primarily it is connected, its main connection is with the other area that is analogous to it or homologous to it on the other side, exactly like the mirror image of it. So now, this area, Broca's, enables us to process syntax and the lexicon and grammatical, words, and to activate speech, program and activate for speech. The other area here enables us, when we speak, to put the appropriate tone of voice into what we say so that we become expressive, that we color our words with our emotions. How you say something in many different ways based on the tone of your voice, that is a function of that area here. So the same for an area here called Wernicke's area. That area enables us to recognize speech, to comprehend speech. Across from it on the other side, there's an area that it's mirror image, but it is not called Wernicke's area because language basics are focused in the left hemisphere. So the other area, analogous or homologous to Wernicke's, enables us to process the tone of voice of someone else's speech. So we'll be able to understand if they are sarcastic, if they are happy, excited, if they are angry, and so on. So these networks are specialized, the networks that are specialized in complex cognitive functions, they span multiple brain regions, like we call them the association cortices. The association cortices that associate, for example, the frontal cortex with the posterior, associate your decision making, your working memory, your attention skills with your vision, for example, with your auditory systems, and so on. So these complex cognitive functions, they require the association cortices because these cortices integrate multiple inputs from different cognitive, perceptual, sensory modalities. Now we look at the functions. We look at now we have the anatomic landmarks, and the anatomic landmarks, when they start to work, they give us behaviors, they give us functions. So we look at the functional networks now, and they go parallel in sync with the anatomic networks. So first, we have immature forms of sensory motor, visual, and auditory networks. Sensory motor means you move something and you feel it as it moves. You know if you are, for example, going too far, or if you are going, there's so much pressure that you put here and there. So monitoring your movement and getting sensory feedback about how you are moving in space. So these sensory motor functions and visual and auditory functions, they are detectable by the 30th conceptual week. So after conception, in week 30, then we have these functional networks that mediate sensory motor skills, visual and auditory functions. And remember, I mentioned before that, for example, that the middle ear and the inner ear are formed adult size, and that is by week 20-21. So it's about 4 months or so, and then you have the mechanism to hear is there, but you need it to connect with the cortex. And what we are speaking about now is what's happening in the cortex here, the areas of the auditory system or the visual system and so on, that make sense of these signals that rise and reach there. So even though, for example, the auditory mechanisms develop, the peripheral system develops by 4 months, then way later you are going to have the cortex now starting to kind of actually to connect and give you functions. And this is where you can test the baby and they would show evidence, for example, that they can hear, they can distinguish the mother's voice from other voices, the mother's language from other languages, and so on. So these networks demonstrate adult-like topography, means the basic architecture of these networks is present at birth. And there are minimal changes during the first postnatal year in terms of the general architecture, the general layout. But we are not saying that there is no change. It is the overall. But of course, the change is robust in terms of the cellular level. The dorsal attentional network and the default mode network, they begin to form after the primary networks are fully in place. What does that mean? We said basic functions like auditory functions, visual, sensory motor functions, these are primary, elementary functions. Then dorsal attention, attentional mechanisms. They have frontal parts of the brain that mediate attention, but you also need dorsal. You need function, areas here that are also activated, and also on the dorsum or the back of the cortex. So basically frontal and parietal here. They connect, and there are circuits that mediate attention. So these develop after you get the auditory, and you get the visual, and you get the other primary sensory functions in place. Then also the default mode network. The default mode network was basically discovered by accident in 1997. And what it does is it has many functions. It basically enables you to have introspection, to look deeper into yourself. It enables you, it is the mechanism that is very heavily involved in daydreaming. It is the mechanism that part of it enables you to kind of translate, or to be aware of yourself, in like reflect inward, and to be aware of yourself and your emotions, and kind of evaluate information to have, so that you can have introspection or intuition. This mechanism, the default mode network, is critical for creativity, and it works primarily when you are not working on goal-directed behavior. If you are not sitting, say, in a class listening and engaged and kind of have a goal somewhere and focusing on your goal, that mechanism, the default mode network, immediately starts to be activated. And it pulls things from your subconscious level to your conscious level, and highlights them and makes them kind of, hey, red flag here, you haven't done this for a long time, you should do this, you should do that. And it keeps kind of bringing things and making connections that you are not aware of, and then highlighting these. But it only is activated when you are not focused on goal-directed behavior. It was accidentally discovered when a researcher with the name, I forgot his first name, but Raquel, he was neuroimaging someone, and then as in between tasks, he just kind of forgot to, and left the imaging machine on. And then he was doing something, and he came back. There wasn't supposed to be any activity because he wasn't telling the person to do anything. And then he looked and found the thing so activated. And then he then researched that after, he discovered the default mode network. And I have his first article. If anyone is interested, I'll be happy to share it. That is when he first discovered it and published it. Oh, what happened there? Oh, I see. Oh, I was concerned that the system might have malfunction, but it's okay. So here's just to give you a taste of different networks. For example, here you can see the attentional network, the green here, the dorsal. It connects the prefrontal cortex and the frontal part of the brain with the parietal area here, and also with the parietal, and also with the somatosensory areas and some motor. Because attention also connects with motor, and this is where you have the hyperactivity, for example, and so on. You can explore, but I'm like, just to give you a taste. I'm not asking about the identification or the anatomy of these things. So the maturation of the physical networks themselves and how they connect, and the sequence of their connections, is correlated with the functions, simply because if you don't have the circuit, you are not going to have functions. So you build the circuit first, you build the engine, and then it will work. It will give you functions. So the completion of the visual, auditory, and sensory motor networks give rise to developmental milestones in visual, auditory, and sensory motor functions. And the connectivity between the thalamus and the salient network at age 12 months gives rise to the development of working memory functions. What is the salience network? This is basically a network that enables you to have cognitive control over attention, and it is driven by motivation. So if you are motivated for something, you feel that there's salience, that this is important for you. If you are motivated, you focus more, you deploy more cognitive resources, and you recruit functional networks to do the focusing and the processing so that you can get that information about the thing that you are motivated to learn or to know about. Children very young, of course, they know if what you are saying has value or if it doesn't. And children, they don't control their behavior as well as adults. And the strategy we can derive out of this is, since children very young, at a very young age, 2 years and so on, they are able to understand the value of the information that they are receiving. And that is going to make that, they will be able to determine if that is salient to them, that is important or not for them. That is going to influence their behavior. So in the classroom, whether in a preschool or beyond, if the child is hearing information that is way above their head, they cannot relate to that because their level is way below for whatever reason, then they are not there. They determine that this doesn't connect with anything that I have. It is not salient to me. It doesn't matter to me. And if it is something they already know, and then you are kind of saying it and repeating it, you are not challenging them, you are not giving them new information, they are going to determine, oh, I know it already, why should I care? So this is where we need to focus, we need to identify the exact area where you are challenging them right, so that you do not give them, a lot of the lessons and stuff that you plan that is all information that they know, or it is way far. So you need to find that Goldilocks kind of level where it is just right. And how do you do that? It is by understanding your student, by understanding how the student actually learns, how they think, and by being invested in learning more about them. And that is going to enable you to find that comfort zone where you are not going to challenge them too much. You are not going to kind of not challenging them. You just challenge them to a level where they are comfortable enough to process what you are giving them, and to have something new that they can acquire. So I hope that as you go through the material, consistently, constantly ask yourself, how do I translate this into a strategy that I can use in my clinical practice or in my teaching in my classroom? So working memory, you as educators, or we as educators, know that working memory is very important for retrieving information and for taking in information. So it is the kind of skill that enables us to feel ourselves thinking. So when you are thinking and speaking to yourself inside of your mind, that is working memory in action. When you are reading silently, that is working memory that makes it possible for you to hear yourself as you are reading. So the working memory skills, there's a stage when children are still building. This is just beginning, but children are still building working memory, and they don't have much kind of a span there. They play and they verbalize. They play and even by themselves, and they speak. That is a reflection of their thought process. As they are speaking out loud, this is how they think in their mind. But they don't have the ability yet, the frontal part of the brain is not mature yet, to make them think inside of their brains. So Vygotsky spoke about this, and he named it something like self-talk, and he spoke about how it is internalized around age 4 years. So by 4 years, the child has now built quite a robust working memory skills, and based on that, they are now able to use that working memory function to think inside of their head instead of speaking as they are verbalizing. And if you are attuned to that, and you look at the children as they grow in the preschool, for example, and you can use it as an indicator of how their working memory is developing. Or even adults, when they start talking to themselves and stuff, that is because working memory has decreased, and they have to refresh their attention by repeating things, by also refreshing their working memory by saying things to themselves and keeping these things refreshed. You hear it inside, you say it, and that makes you kind of keep rehearsing it. So again, these are things that we can look and predict the functioning of a child or adult based on. These are some studies that kind of give us representations of brain connectivity after birth on a lower scale, and of course these connectivity patterns are much richer and much more sophisticated and complicated, as we age, as we grow. So at birth, the adult-like activation patterns, what we're speaking here is about the pattern. We're not speaking about how robust or how extensive, no, but just the pattern of how it is progressing. These are present at birth. For example, tactile and proprioceptive stimulation induce similar firing patterns. We know tactile has to do with touch and pressure and anything that has to do with that sense, tactile. And proprioception is the sensation that we get, or the information that we get from the joints of the body and the muscles of the body. And as we move in a certain way, for example, we get the proprioceptive feedback to tell us, oh, I am lifting my tongue, I am lifting my tongue and shifting it to the right, to the left, I'm rounding my lips. You feel your organs as you are moving them around. You feel your body in space. So this is all proprioceptive proprioception. And some occupational therapists and physical therapists sometimes might call it the sixth sense. So again, it comes from muscles and joints, and it gives you information about how these muscles and joints are performing the movements that you want them to perform. The Auditory also and olfactory and visual inputs are also going to activate to induce adult-like activation patterns. At that age, about 2 to 3 months of age, then you have lateralization of auditory and language functions. That means the left side of the brain has language basics. For example, syntax, lexicon, comprehension of speech, and so on, basics in the left hemisphere. We are not saying that the right hemisphere is not needed. It is that the brain localizes functions in different areas so that you don't have redundancy, so that you have multiple areas, each one is doing a function, and then you integrate and you use this and that, and you can do more with less. So lateralization or lateralized activation, for example, when you hear language, you are going to have more firing in the left hemisphere. And also, like sound processing and so on for speech, particularly for speech. But if you hear music, it's going to activate left and right hemispheres, for example. So that lateralization is, emerges at that time, 2 to 3 months, and in response to native language stimulation, like when the mother is speaking or the father is speaking, now the baby understands the native language and the basic sounds. Now they are going to activate left hemisphere, and but if it is a foreign language, it's a whole different matter. So but the activation patterns follow the same pattern of the adult brain. For example, when you hear a sound, say a word, first you have the posterior temporal regions activated. So here, when we say posterior temporal, this is the temporal cortex. Posterior temporal is the area that is known as Wernicke's area, and that recognizes speech and is specialized in comprehension of speech and assembling sounds and so on. So the signals are first recognized here, and then you are going to go to the auditory cortex, is activated here next, and then the temporal pole, this area here, is also activated, and then you have activation of the in this area called the angular gyrus, and then you go to Broca's area, which is 44 and 45 activated after, because you need, for example, you hear the word, you recognize it as speech, you assemble the sounds of the word, and then you go to behind this surface, you go to the auditory association cortex and say, give me the meaning of that word. Say, for example, C-A-T. And then it communicates with the hippocampus and says, what does that, do you have anything that looks like, cat? What does it mean? And then that hippocampus is going to get you all information about the word cat. And then now you assemble cat, the sounds of cat, with the meaning of what cat means, and then send the information to the parietal cortex here, angular gyrus, and send it finally to Broca's area to say, is cat a noun? Is it a verb? Do you put it at the beginning of a sentence? Do you put it at the end? How do you place it in syntax? And let's put it in a sentence, and so on. And Broca's area keeps communicating with Wernicke's, and Wernicke's with Broca's, and also the areas here in the sensory cortex, so that you feel and you imagine the texture and feel of something that you are speaking about. And also the visual cortex is also connected, so that you can a word can make you see images in your mind, in your head. So that is the normal pattern that you and I experience when we hear a word or for example, language. And that same pattern is followed by children, given that they are typically developing. So the neonate, 2 to 3 months old, exhibit signs of early learning. And for example, even before that, the baby cries 2 weeks, and then you come in, the baby learns to stop crying. And then eventually, in days, they are going to learn, oh, I want to call someone to come and help me. They will start to cry. You come, they even hear your feet come and shuffle, and then they stop. So that is learning. And they can also explore the looking and taking turns. So much is happening. It is all learning. But the basic signs anyone can recognize. 2 to 3 weeks, there's some intention and purposefulness to what they do. 2 to 3 months, the baby is able to, again, that is a skill that they have when they are born. They distinguish speech of other, native language, their mother's voice, and so on. The 2 to 3 months, they are able to distinguish between speech and music. When they hear the mother's voice, they have an activation pattern that is different when they hear the voice of a stranger. So they activate, when they hear the mother's voice, they activate language centers and emotional processing centers. And they really pay attention a lot to the tone of your voice and the expressions of your face and so on. So then let's speak for the next few minutes about risk factors for altered functional networks. We spoke about this. You did your own search, and this one is going to add more to what you already know. So there are risk factors that will cause abnormalities in circuit formation and connectivity. And these abnormalities will lead to behavioral correlates, behavioral problems. So whether they arise from genetic or epigenetic factors, they alter brain circuits. But what we are focusing on here are environmental things that it can really be prevented. I don't know about preterm birth though, but a lot of things can be prevented. So these alterations may result in atypical behaviors, for example, including abnormalities in executive functions or executive functioning. That is your self-regulation ability. That's your ability to develop goals and to pursue these goals. The ability to regulate your work habits, how you interact with others, how you have a goal in life to succeed, academically or anything that you want to do, basically managing your life from A to Z. So also the person will have difficult problems in social and emotional development, and they will have academic performance. Of course, that's going to be affected by executive functioning. They don't know how to manage, should they do homework first? Should they do homework at all? Should they be invested in schooling? And all of these things, and the emotional and social also are going to contribute. So academic performance is going to be affected by many, many things. So these deficits could last, could continue with the person through their lifetime. So let's speak about drug exposure. First, prenatal substance exposure, basically through the mother, may elicit teratogenic effects in the embryonic stage. Teratogenic means it is going to change the formation itself. This is going to lead to anatomic abnormalities. So now the effect can be either direct or indirect. And we are focusing here on network formations or the formation of circuits and how they function. So, but some statistics that are alarming here are that 4.4 to 5.1% of pregnant women use illegal drugs. 16.3% smoke cigarettes during smoking. 10.8% consume alcohol during pregnancy. So all of these, I mean, these basically can be managed. I mean, they can be stopped. And if people know the risk, they will probably do something to minimize it for their offspring. Nicotine and psychoactive drugs that are used, for basically to stimulate and to be high and all of this, they influence monoaminergic, the neurotransmitter systems. So there are neurotransmitters that are called monoamines, or they belong basically to this category. And these include dopamine, epinephrine, and serotonin. Dopamine is the neurotransmitter for motivation, for movement, and for emotional regulation. So if someone has decreased dopamine, they are going to shake, and that is, for example, people who have Parkinson's disease. The major problem is deficiency in dopamine. If you have dopamine but you are not circulating and using it to signal correctly and so on, there is serotonin abnormality with that these are the major things that are causing depression. Epinephrine is what enables you to, we used to call it adrenaline. It enables you to be alert and to be kind of have a certain level of kind of like stress that is good stress to make you, on the alert all the time. So these systems are the systems we use to learn. These are the learning mechanisms. For example, people with ADHD, this is serotonin is a big problem. Dopamine is a big problem because one of them activates the other. Epinephrine. So all of these, if someone has abnormalities in them, they have emotional problems. They are going to have attentional problems. They're going to have behavioral problems. They might have addiction problems. And of course, you understand that there's a high likelihood, of course, that it increases the likelihood of a child picking up the same habit themselves, whether smoking or drug. So these cause alterations, physical alterations in the networks. Nicotine and psychoactive drugs impact neural proliferation. That's if the mother is consuming that during pregnancy. Neural cell migration very early in life. That is devastating, and you just have a little problem and it will grow into a tremendous problem if you have cell migration problems, because that is a foundation, and if you build the house with a weak foundation, then forget it. Axonal growth is going to be influenced, and the axon, if it is not developed well, it's not going to function well. Arborization, the structure of the dendrites that actually receive signals are not going to be normal, whether you are not going to have many and you are not going to have them shaped normally. Also, you add to this reduced nutrition due to placental and umbilical vasoconstriction. So that is going, the umbilical cord will constrict and the placenta will constrict, and as a result, you are not getting enough supply of nutrients to the baby. And that is, it results from this nicotine, and it results from psychoactive drugs. Nicotine, yes, when the woman who is pregnant consumes it, the risk is greatest. But if she is pregnant and she is inhaling nicotine, there will also be an effect on the baby. So that's a very serious matter. Even third-hand smoking now is known to have an impact on you as an adult, brain functions, and now imagine the baby with the developing brain. So third-hand smoking is the smoking that you find when you buy a used car, or you use a car that someone else used that had smoking embedded there in the fabric, or you go to that hotel and say, I would like a non-smoking room, and you get in, and the smell of smoking is in the sheets and the towels everywhere. So that is third-hand smoking. But of course, second-hand smoking is very serious, whether the mother inhales it or she actually is using it herself. The cocaine and marijuana, the impact on functional connectivity, again, hits the areas that are targeted by these drugs. For example, people take these drugs to get activate the amygdala, to activate the medial prefrontal cortex, the thalamus, anterior cortical areas, and these areas. So naturally, they are going to be circulated more. I mean, the substances are going to be circulated more around these areas, and these are serious areas. The amygdala is the emotional processing center, and the medial prefrontal cortex is your decision and working memory and self-regulation center. You know that there are laws, you need to observe, you need to abide by rules, and so on. And now this is affected. Now you can imagine the abnormalities. The thalamus, as I mentioned, it is really critical. It affects almost everything. And the anterior cortical areas around here, they have, then attention could be affected, decision-making, judgment, and basic, very, very important self-regulation. All of these this area is what manages the entire life. Your entire life is in this area. Marijuana especially causes disrupts connectivity of the caudate and the insula. For example, here, the caudate is this structure here that you could here. You could see this structure here is the caudate nucleus, and it has a head and a tail on the other end. So that is very important for movement, for emotions, for learning, for memory. Basic, very important functions for learning all kinds of things. The insula, which is part of the default mode network, and also it is important for speech and for making you feel your emotions in your gut. It enables you to translate your emotions that you are not aware of into feelings that you become aware of. And it translates these feelings into physical responses, like you feel something and immediately you're so flushed and so angry, and your physical, your body is changed in response to your feeling. That is the insula in working with other areas like the amygdala and hypothalamus and so on. So there's abnormalities in how these connect. And you can imagine now, use the question, what kind of strategy will I get out of that? Do you have children now who have emotional problems that are persistent and very young age? Then you ask a question, are they exposed to these things, for example? I know it's a very sensitive thing, but at least what you can observe, based on what you know, you are not going to go around and ask people, but at least if this information comes up or you are aware of, then you know that there is an effect. Of course, one of the things that we don't want to do is to make the parent guilty, because if we know or we do not know, the cause, we have the child that we have to deal with. Also, diffuse patterns of hypoconnectivity and hyperconnectivity between the insula, amygdala, and thalamus. So many areas that some of them have decreased activation, some of them have hyperactivation. The areas that you don't want to have hyperactivation in are the amygdala. For example, people who have phobias, the amygdala is so active and just waiting to be activated at the least thing that you see. The thalamus is, again, it gates sensory stimuli, and when you have these areas disconnected basically, or some of areas are hyperconnected, that is going to lead to overstimulation, hyperactivity, impulsivity. When we look now at depression, depression is genetic. There are genes that are associated with depression. So when the mother's depressed, you have now the stress response activated. It is like someone is fighting, physically fighting. You have the stress response, where you have high cortisol levels in the bloodstream, and that raises your blood pressure, and it makes you under stress, and it is going to have an impact on learning and emotions, all kinds of things, and how the mother interacts with the baby, and so on. The medications also in themselves, they put the child at risk. So these lead to disturbances in connectivity of the brain and the formation of circuits and how they function. Severe maternal depression during the second trimester causes stronger functional connectivity of the amygdala, and that means that you have overstimulation of the emotional system, emotional reactivity. Constantly, the person is active, the person is so kind of, they can't regulate their emotions. The child might have a temperament problem, and you wouldn't take much to agitate them or to get them to, to cry and to be, and to act up. Disturbances in the limbic system, and the medial prefrontal area. So emotional system is significantly impacted. The learning system is significantly impacted because they are limbic, and the prefrontal cortex, that is your decision making, your judgment, your law abiding, the thing that makes you regard rules and become a law obedient citizen. All of that is going to be affected. So these children are gonna need some serious work, but you have to kind of understand again how to get to service them best. And if you learn more of the case, you'll be able to help them even better. Depression medications itself or themselves, they are known to lead to widespread reduction in the white matter, in the maturation of the white matter. There will be lower interhemispheric synchronicity, where you want the left hemisphere to work in sync with the right hemisphere. For example, when it comes to language function, you hear a word like an expression, it's raining cats and dogs. So you hear the word, it is raining. You hear the actual words. They go first to the left hemisphere. You understand, and you understand where to put the words and all of this, where everything fits. And immediately you want to, of course, get the meaning. The right hemisphere is going to, while you are working on assembling and getting this done, the right hemisphere immediately is going to tell you the meaning. It says, suppress your interpretation here. Because you are not going to find it in the words. I am going to tell you what the whole thing means. So it tells you, oh, it doesn't mean that you look for cats and dogs coming down from the sky. It means blah, blah, blah. So both of these have to work in sync together. And if there's a discrepancy, then you would not be able, for example, while you are processing, you will not be able to understand the tone of voice and what it means, for example, and you might miss so much in terms of social, or misunderstand a lot of social cues and that we transmit through our voices. There are also shorter, what is known as interburst intervals. That is shorter periods of relative rest in the brain activity, like kind of you fire and then you kind of decrease firing. These periods are shorter than normal. That means there is hyperactivity there. The brain is just constantly going on and on, and that is going to produce many behavioral problems. That may lead to poor temperament, may lead to impulsivity, and it may lead to hyperactive behavior. So preterm birth is the last thing that I'll speak about. I am looking, and the time is, the tape is getting longer. So at the time in preterm birth, when that occurs, if it's 2 or 3 or 4 weeks, for example, or more, the brain, the neural circuits are still forming. Synaptogenesis is ongoing. Neural cell migration is still ongoing. Axonal growth is ongoing. All of these processes are ongoing, and the baby was supposed to complete that before being born. Now, when the babies are born prematurely, they spend some time in the first whatever time that they spend after birth. Say, for example, if they are born 4 weeks prematurely, they are going to spend that time, the 4 weeks, forming what they didn't form inside. So they are going to experience a 5-fold increase in cortical surface areas and doubling of the cortical curvature, going up and down, convolutions. And also the time within the kind of time frame that coincides with their 3rd trimester of pregnancy. So you're going to have to make that up outside. But now the question is, if the baby who didn't experience any problems, and they have all that, the structural components in place, and they are ready to function at birth, they don't have to wait for anything. If the other babies who are preterm, they spend the first 2 to 3 months simply catching up, what capacities will they have to be able to continue to receive stimulation and to build networks and to do all of that? So that is something that you need to think about. And think also about the meaning of 1 day in the life of a baby. That 1 day could equal probably 1 year in the life of an adult. So how much of what are the consequences of losing that period of time when the baby is born, and their brains have to simply kind of focus on kind of continuing the building process, and they don't have yet the time to benefit from as much from the stimulation that is that the environment is supposed to provide. That's a question. I don't know if we can have the answer to it, but it's just an interesting question. Preterm birth is known to affect the quality of brain circuits and how they form. And however, the overall layout of the networks is the same, but it's just how these networks are formed in general and how they function. these babies are highly susceptible to neurodevelopmental deficits. There's a lot of research speaking about premature birth. So basically, children who are born preterm, they experience impaired brain development in the following areas. They have decreased brain volume. They have decreased cortical gyrification. The gyri are not well developed. And they have decreased maturation in the gray matter and the white matter structures. They have pervasive disruption in the thalamocortical connectivity, that is the first network that appears, and also the structural and functional deficits, means the anatomy and the function are impacted. There's decreased interhemispheric connectivity, and there's impaired lateralization of language. We have to keep in mind that these deficits are going to be correlated with the severity of the problem. For example, someone who is born 8 weeks preterm is not going to have the same, the problems will not be as severe as when someone is 4 weeks preterm, and so on. If it's 2 weeks preterm versus 4, so the longer the period that the child spends, outside of the womb when they are not supposed to spend that, is going to have to determine the severity of these problems that we are speaking about. So these alterations may lead to many behavioral consequences, including cognitive delays, decreased work and memory functions, executive control executive functioning deficits that are associated with ADHD and autism and so on, and autism and ADHD. So in conclusion, just to wrap things up, brain circuits begin forming prenatally and they continue robustly after birth, and they continue through the postnatal years. The process at the beginning is driven by genetic programming, but at birth, then you have the epigenetic factors shaping and influencing the formation of these networks, and the baby is born with these tentative networks, but environmental stimulation is going to determine how they are dismantled or how they are strengthened. Long-range circuits form before local circuits, and epigenetic processes exert major influences on circuit formation and performance. For example, there are many major risk factors, including maternal depression, maternal use of drugs and alcohol, and then nicotine exposure and preterm birth. So with this, now I stop, and you have the tape. You can go back to as many times as you like. So, and thank you for your patience.